Hello guys, welcome to my YouTube channel. Today's our topic is the steps involved in the genetic engineering. What is genetic engineering? Genetic engineering that we can call it as a gene cloning. Then we have to know the gene cloning. Gene cloning involves separation of a specific gene. We have to separate the specific gene or a DNA fragment from a donor cell. मतलब we have to separate the target DNA fragment, then attaching it with a small carrier molecules, that's a vector, then replicating this recombinant vector into a host cell. Now let's discuss about it. We have to take the specific gene or DNA fragment attaching with the vector, then we can form a recombinant vector or recombinant DNA. That's what we have to replicate in host cell, that we have to discuss. Then there is the several steps for the gene cloning. First steps in the isolation of the target gene, then selection of a suitable vector. We have to take a suitable vector that we have to select from here, and incorporation of the target DNA fragment into a vector. We have to mix the both DNA fragment that we have to taken, and we have taken a suitable vector that we have to mixed both together. Then transformation of the recombinant vector into a suitable host cell. Then the fifth step is isolation of the recombinant host cell. We have to isolation of the recombinant host cell. Let us discuss the diagrammatic form of the 5 methods. This is the human DNA containing gene of interest that we can call the target DNA fragment. The target DNA fragment, this is the target DNA fragment. We have to isolation of the target DNA fragment. Then we have to take a suitable vector, that is we can take the plasmid vector as a suitable vector, then selection of the plasmid vector, then we have to isolate the plasmid vector from there, and we have to isolate the human DNA or isolation the DNA fragment from here. Then we have to mix both of them together. We have to cut the vectors in a specific site by the restriction endonuclease enzyme, and we have to cut the same DNA fragment by using the restriction enzyme, and we have to attaching both together. Insert human DNA into the plasmid. We have to take the DNA fragment and we have to attach in the vector. Use the DNA ligase for attaching the both DNA fragment and the vectors, plasmid vectors. Get the recombinant vector. Then we have to replicate it in the host cell. This is the host cell, and we have taken the vectors in the host cell and we have to transformation of them. So what is the fifth step? Isolation of the recombinant host cell. We can, by cloning only, we can got the isolation of the recombinant host cell. Let us discuss about all the points. Isolation of the target DNA fragment or the gene. At first, donor DNA fragment should be isolated. What is the donor DNA fragment? This is the donor DNA fragment or the target DNA fragment that we have to isolate it. There are two methods for the isolation of the desired gene or DNA fragment. We are isolating the DNA fragment or target DNA. There is two methods for the isolation. First one is using the restriction endonuclease enzyme, and second one using the reversible transcriptase enzyme. In case of the restriction endonuclease enzyme, the enzyme restriction endonuclease is a key enzyme. It is a molecule. What it do? It cut the DNA at a specific restriction site of the DNA for its action. It cut the DNA at a specific site of the target DNA. Let's second method. using the reverse transcription enzyme. It synthesizes complementary DNA strand. Complementary DNA strand we have to synthesize of the desired gene by using its mRNA. Let us second point, selection of the suitable vector. We have to take a suitable vector for their action. So what is the suitable vector? We can take a suitable vector as a plasmid vector. Plasmid vector is a suitable vector. Before the introduction of the donor DNA fragment into the host cell, a suitable vector should be selected. What purpose we are taking the suitable vector? That we are taking the DNA fragment as a vector and the recombinant. We have to form the recombinant vector that can replicate. That's why we are taking the suitable vector that we can mark. Cloning vector is a DNA molecule. Cloning vector nothing but a DNA molecule capable of the self-replication inside the host cell. What's the function of the recombinant vector? It's replicate the inserted DNA fragment inside the host cell. For example of the cloning vectors, plasmid vector, expression vector, but point. Incorporation of the donor DNA fragment with a suitable vectors. We are taking the suitable vector as plasmid vector, that's why we are directly taking the plasmid vector. The plasmid vector is cut. We have to cut the plasmid vector from here, plasmid vector, and open. Cut open by the restriction endonuclease enzyme for the isolation of the DNA fragment. Okay. Donor DNA fragment and the plasmid vector are mixed. Both are mixed. Previously, I am saying that the DNA fragment and the vector we have to mix for the formation of the recombinant vector. That only they are mixing both together. In the using DNA ligase for the mixing of both together. That's only. And transformation of the recombinant vector into the suitable cell. We have to transformation the recombinant vector into the host cell. Then the recombinant vector is transferred into the suitable host cell. That's host cell are, we have taken the E. coli host cell. Here the example Bacillus, Helicobacter and Pyroli are the naturally component we have taken. Those bacteria are not naturally competent. Their recombinant vector are incorporated by the artificial method. What are the artificial method? Calcium ion treatment and the electroprotonation. It is. Then the last method, isolation of the recombinant host cell. We have to isolate the recombinant host cell. The recombinant host cell is then grown in culture media. We have to use the culture media for the recombination. Then we have taken culture media for the cloning. But the culture media may contains the colonies. That culture media contain the colonies, both recombinant cell and as well as the non-recombinant cell. For the isolation of the recombinant cell, we have to isolate the recombinant cell. For the isolation of the recombinant cell from the non-recombinant cell, marker gene of the plasmid vector is employed. Naturally, we are isolation the recombinant cell, but when we are taking the non-recombinant cell, from the non-recombinant cell we have to isolate the recombinant cell. At a time we are using the marker gene of the plasmid vector in that. What are the examples of the marker gene? Is PBR322 plasmid vector, ampicillin resistance gene and the tetracycline resistance gene as a marker.